Burgess Shale. The Burgess Shale is a fossil-bearing deposit exposed in the Canadian Rockies of British Columbia, Canada. It is famous for the exceptional preservation of the soft parts of its fossils. At 508 million years old Middle Cambrian, it is one of the earliest fossil beds containing soft part imprints. The rock unit is a black shale and crops out at a number of localities near the town of Field in Yoho National Park and the Kicking Horse Pass. Another outcrop is in Kootenay National Park 42 km to the south. History and significance. The Burgess Shale was discovered by paleontologist Charles Walcott on 30 August 1909, towards the end of the season's fieldwork. He returned in 1910 with his sons, daughter, and wife, establishing a quarry on the flanks of Fossil Ridge. The significance of soft-bodied preservation and the range of organisms he recognized as new to science led him to return to the quarry almost every year until 1924. At that point, aged 74, he had amassed over 65,000 specimens. Describing the fossils was a vast task, pursued by Walcott until his death in 1927. Walcott, led by scientific opinion at the time, attempted to categorize all fossils into living taxa, and as a result, the fossils were regarded as little more than curiosities at the time. It was not until 1962 that a first-hand reinvestigation of the fossils was attempted by Alberto Simonetta. This led scientists to recognize that Walcott had barely scratched the surface of information available in the Burgess Shale, and also made it clear that the organisms did not fit comfortably into modern groups. Excavations were resumed at the Walcott Quarry by the Geological Survey of Canada under the persuasion of trilobite expert Harry Blackmore Whittington, and a new quarry, the Raymond, was established about 20 meters higher up Fossil Ridge. Whittington, with the help of research students Derek Briggs and Simon Conway Maurice of the University of Cambridge, began a thorough reassessment of the Burgess Shale, and revealed that the fauna represented were much more diverse and unusual than Walcott had recognized. Many of the animals present had bizarre anatomical features, and only the slightest resemblance to other known animals. Examples include Opabinia, with 5 eyes and a snout like a vacuum cleaner hose, and Hallucigenia, which was originally reconstructed upside down, walking on bilaterally symmetrical spines. With Parks Canada and UNESCO recognizing the significance of the Burgess Shale, collecting fossils became politically more difficult from the mid-1970s. Collections continued to be made by the Royal Ontario Museum. The curator of invertebrate paleontology, Desmond Collins, identified a number of additional outcrops, stratigraphically both higher and lower than the original Walcott quarry. These localities continue to yield new organisms faster than they can be studied. Stephen J. Good's book Wonderful Life, published in 1989, brought the Burgess Shale fossils to the public's attention. Good suggests that the extraordinary diversity of the fossils indicates that life forms at the time were much more disparate in body form than those that survive today, and that many of the unique lineages were evolutionary experiments that became extinct. Good's interpretation of the diversity of Cambrian fauna relied heavily on Simon Conway Morrissey's reinterpretation of Charles Walcott's original publications. However, Conway Maurice strongly disagreed with Good's conclusions, arguing that almost all the Cambrian fauna could be classified into modern-day phyla. The Burgess Shale has attracted the interest of paleoclimatologists who want to study and predict long-term future changes in Earth's climate. According to Peter Ward and Donald Brownlee in the 2003 book The Life and Death of Planet Earth, climatologists studied the fossil records in the Burgess Shale to understand the climate of the Cambrian explosion. It can be used to predict what Earth's climate would look like 500 million years in the future as a warming and expanding sun, combined with declining CO2 and oxygen levels, eventually heat the Earth toward temperatures not seen since the Archean Eon 3 billion years ago, before the first plants and animals appeared. This in turn furthers understanding of how and when the last living things on Earth could potentially die out. See also Future of the Earth. After the Burgess Shale site was registered as a World Heritage Site in 1980, it was included in the Canadian Rocky Mountain Park's WHS designation in 1984. In February 2014, the discovery was announced of another Burgess Shale outcrop in Kootenay National Park to the south. In just 15 days of field collecting in 2013, 50 animal species were unearthed at the new site. Geological setting. The fossil-bearing deposits of the Burgess Shale correlate to the Stephen Formation, a collection of slightly calcareous dark mudstones about 508 million years old. The beds were deposited at the base of a cliff about 160 m tall, below the depth agitated by waves during storms. This vertical cliff was composed of the calcareous reefs of the Cathedral Formation, which probably formed shortly before the deposition of the Burgess Shale. The precise formation mechanism is not known for certain, but the most widely accepted hypothesis suggests that the edge of the Cathedral Formation reef became detached from the rest of the reef, slumping, and being transported some distance, perhaps kilometers, away from the reef edge. Later reactivation of faults at the base of the formation led to its disintegration from about 509 million years ago. This would have left a steep cliff, the bottom of which would be protected from tectonic decompression because the limestone of the Cathedral Formation is difficult to compress. This protection explains why fossils preserved further from the Cathedral Formation are impossible to work with. Tectonic squeezing of the beds has produced a vertical cleavage that fractures the rocks, so they split perpendicular to the fossils. The Walcott Quarry produced such spectacular fossils because it was so close to the Stephen Formation. Indeed, the quarry has now been excavated to the very edge of the Cambrian cliff. It was originally thought that the Burgess Shale was deposited in anoxic conditions, but mounting research shows that oxygen was continually present in the sediment. The anoxic setting had been thought to not only protect the newly dead organisms from decay, but it also created chemical conditions allowing the preservation of the soft parts of the organisms. Further, it reduced the abundance of burrowing organisms. Burrows and trackways are found in beds containing soft-bodied organisms, but they are rare and generally of limited vertical extent. Brine seeps are an alternative hypothesis. See Burgess Shale type preservation for a more thorough discussion. Stratigraphy. The Burgess Shale Formation comprises 10 members, the most famous being the Walcott Quarry Shale member comprising the Greater Philippid Bed. Taphonomy and diagenesis. There are many other comparable Cambrian lagerstätten. Indeed, such assemblages are far more common in the Cambrian than in any other period. This is mainly due to the limited extent of burrowing activity, as such bioturbation became more prevalent throughout the Cambrian. Environments capable of preserving organisms' soft parts became much rarer. The Precambrian fossil record of animals is sparse and ambiguous. Biota. The biota of the Burgess Shale appears to be typical of Middle Cambrian deposits. Although the hard part-bearing organisms make up as little as 14% of the community, these same organisms are found in similar proportions in other Cambrian localities. This means that there is no reason to assume that the organisms without hard parts are exceptional in any way. Many appear in other lagerstätten of different age and locations. The biota consists of a range of organisms. Free-swimming nectonic organisms are relatively rare, with the majority of organisms being bottom-dwelling benthic, either moving about vagrant or permanently attached to the seafloor sessile. About two-thirds of the Burgess Shale organisms lived by feeding on the organic content in the muddy sea floor, while almost a third filtered out fine particles from the water column. Under 10% of organisms were predators or scavengers, although since these organisms were larger, the biomass was split equally among each of the filter-feeding, deposit-feeding, predatory, and scavenging organisms. Many Burgess Shale organisms represent stem group members of the modern animal phyla, though crown group representatives of certain phyla are also present. Working with the Burgess Shale. The fossils of the Burgess Shale are preserved as black carbon films on black shales, and so are difficult to photograph. However, various photographic techniques can improve the quality of the images that can be acquired. Other techniques include backscatter (SEM), elemental mapping, and camera lucida drawing. Once images have been acquired, the effects of decay and taphonomy must be accounted for before a correct anatomical reconstruction can be made. A consideration of the combination of characters allows researchers to establish the taxonomic affinity.